We want to look at the integumentary system now. And what about that? The epithelium, the epidermis, and then the dermis also. So as you're talking about the skin, epithelium, epidermis, there'll be a description of the receptors as well. We have to talk about receptors as well. And the first thing is epithelium or epidermis, skin epidermis. Let us look at a histological picture. There'll be 5 layers in the skin epithelium. And what are the 5 layers? What is this C for? Stratum corneum, having some keratin fibers. So there will be a stratum corneum having some keratin fibers. Yes, stratum corneum. What is number 2? What is this G for? This number 2, the G is for stratum granulosum. Stratum granulosum. It has some keratohyalin granules. Keratohyalin granules. But remember, if you have taken a specimen from thick skin, then you will have a middle layer here. What is this? What is this middle layer? That middle layer is called as the stratum L. What is that? Stratum lucidum. So you are telling that if the sample was from a thick skin, could be, palm, or could be the sole, you have a stratum lucidum also? Yes. So which is the first layer then? Again, stratum corneum, C. And what is the second layer? L is stratum lucidum if it is thick skin from palm and sole. Otherwise, generally it is missing. And third layer is G, stratum granulosum, having some keratohyalin granules. What is layer number 4? Stratum spinosum. This is stratum spinosum. Now, if you say that is the stratum spinosum, why would you call it a stratum spinosum? That we will discuss, but the last layer is stratum basale or the stratum germinativum. So this is stratum basale or stratum germinativum? Yes. Stratum basale or stratum germinativum. Now, once you have identified the various layers of skin, we need to discuss more, and then you can draw the diagram also. So what do you want to know more about this? Actually, this is the first stratum corneum, having a lot of keratin fibers. And then what is deep to that? The granular layer with the keratohyalin granules. And what is deeper to that? The prickle cell layer. What is prickle cell layer? That is stratum spinosum. Stratum spinosum. And what is the basal layer having? Actually, basal layer we have to focus upon because there is a cell which is here. What is this? Merkel cell. It's a receptor for light touch sensation. You are holding the pencil, light touch sensation. Receptor is Merkel cell. So you will see the Merkel cell in the basal layer. Yes. And along with that, we will also see stem cells. So as you're talking about stem cells, the stem cells are there in the basal layer. Yes, because they keep dividing and adding more layers from below. There is one more cell in the basal layer, and that we are calling as the melanocyte. So 3 cells basically. One is Merkel cell, which is for light touch sensation, like you are holding a pen, light touch sensation. Then there is a stem cell which is adding more and more layers from below, and then there is a melanocyte forming melanin to counter the sunlight. These 3 cells are there in the basal layer. What about the Langerhans cell then? Actually, Langerhans cell is in the upper layers, and it is antigen presenting cell. If you say it is antigen presenting cell, then what is it presenting? Actually, in the skin you can have bacteria, and it will pick the bacterial components and present it to the lymphocytes. So antigen presenting cell is monocyte derived cell. Actually, monocytes in the bone marrow, they will come to the skin and work as Langerhans cell. They are monocyte derived phagocytic type of cell, and they are antigen presenting cell in the upper layers. Now, once you have this orientation, let's make the diagram ourselves. And as you're talking about the 5 layers of the skin now, what are the 5 layers? The very first layer is stratum corneum. And how do you know that it is stratum corneum? Because I have lot of keratin fibers. So you have a lot of keratin fibers, so this is stratum corneum. Yes. And what is the layer number 2? Stratum lucidum, which is not generally seen. If you see it, then you must have taken the specimen from some thick skin like sole and palm. What about the layer number 3? It is stratum granulosum. And why would you say stratum granulosum? Because it has granules. And what are those granules called? They are called as keratohyalin granules. What is the importance of keratohyalin granules then? They are the one which will form the keratin fibers for the first layer. So there is some keratohyalin granules in the stratum granulosum which are going to form the keratin fibers for the first layer. Yeah, that's what. And what is the 4th layer? 4th layer is stratum spinosum. It is also called as the prickle cell layer. But why would you say prickle cell layer stratum spinosum? Because the cells are having spiny appearance. Prickle cell layer, prickle means spiny appearance. But why these cells have spiny appearance, stratum spinosum, prickle cell? Because during preparation the cells have separated. And normally they were attaching to each other by desmosomes. So when a cell attaches to other cell by desmosomes, there will be some stretching of the membrane and they appear like having some spines. Spines. So stratum spinosum having some cells with spiny appearance, and because of some desmosomal, it is the stratum spinosum which is involved in pemphigus vulgaris. There are some autoantibodies destroying the desmosomes. So this layer will be involved. Pemphigus vulgaris, when there are some skin blisters, there are some autoantibodies destroying the desmosomes, and this will happen. This is stratum spinosum which is involved in pemphigus vulgaris, some autoantibodies destroying desmosomes. The intercellular separation will be there. Intercellular separation can be seen in this problem. And what about this layer number 5? Layer number 5 is stratum germinativum, which is also called as stratum basale. And there are some cells to discuss, like we have this Merkel cell. What about Merkel cell? It is for light touch sensation. It's a receptor for light touch sensation in the basal layer. One more, stem cell. What about stem cell? It is adding more and more layers of skin from below. So this is stem cell, yes. And there is Merkel cell, yes. I think there is one more. Yeah, that is the melanocyte. So what is melanocyte doing here in the basal layer? Forming melanin to counter the sunlight. Now remember that melanocytes are derived from neural crest cells. It is the neural crest cells which form melanin. Earlier it was mentioned that neural crest cell only will form the Merkel cell, but that theory is no more valid. So earlier we used to say that neural crest cell form melanocyte and they also form Merkel cell, but this theory is not evident. And now what is established? That Merkel cell come from surface ectoderm as the skin epithelium coming. So all these cells, where would they come from? Surface ectoderm. It is a surface ectoderm which will form the skin epithelium, and all these cells will come including the Merkel cell also. When you say layer number 4, 5, which is stratum spinosum and germinativum, together they'll form the stratum malpighi. This is a question coming, malpighi. So when you're talking about stratum malpighi, then it is actually 2 layers. That is the stratum spinosum plus stratum germinativum. Okay, this is about the skin epithelium. Receptors. Now we want to talk about receptors. Like which one? Merkel and Meissner and Pacinian and Ruffini. They're also coming as questions. So if you are talking about Merkel cell, it is in the basal layer for light touch sensation. If you're talking about the Meissner corpuscle, then it is actually Meissner corpuscle is this one. This is Meissner corpuscle. It is for 2-point discrimination. If you're talking about Pacinian corpuscle, it will carry pressure and vibration. And if you're talking about Ruffini receptor, it is for the dermal stretch. Dermal stretch. This is all what we want to discuss by drawing a diagram ourselves. This is our diagram. Remember, Merkel cell, as I told you, that is for carrying the light touch sensation, is also important for reading Braille. Braille is read by blind people. They train themselves to read Braille, and a question came to read the Braille, those elevated dots, which receptor is more efficient? And the answer was Merkel, better than Meissner. So Merkel is better than Meissner when it comes to reading of Braille. Actually, we are making a diagram where we are looking at the epidermis and also the dermis. Dermis is superficial and deep. So there will be superficial dermis and there will be deep dermis also. And what is between dermis and epidermis? This is basement membrane. Basement membrane. So this is the basement membrane of epithelium, because basically epidermis is epithelium. And as you remember, in the 5th layer of that is the Merkel cell. So Merkel cell is a receptor which is present in the 5th layer of the epidermis or the basal layer of epidermis, and it is a slowly adapting receptor. That slowly adapting receptor is to carry light touch sensation. Now you will find at the dermo-epidermal junction there is this Meissner corpuscle, and the Meissner corpuscle, which is at the dermo-epidermal junction, obviously it is in the superficial dermis. So Meissner corpuscle, which is at the dermo-epidermal junction, which is in the superficial dermis, what is it for? First of all, the Meissner corpuscle is a rapidly adapting receptor. It is rapidly adapting receptor. And what is it for? 2-point discrimination, tactile discrimination. So 2-point discrimination, in the wards you do this 2-point discrimination, that is carried by the Meissner corpuscle. It is a rapidly adapting receptor. What about the deep dermis? In the deep dermis, I have got this Pacinian corpuscle. Now, when you are talking about the Pacinian corpuscle, which is in the deep dermis, what is it doing in the deep dermis, the Pacinian corpuscle? Remember PP. And what is PP? PP, the Pacinian carry pressure, and not only it carry pressure, but it also carry vibration, and it is a rapidly adapting receptor. Again, this is Ruffini receptor. Now, if you're talking about the Ruffini receptor, is it in the deep dermis? Yes, it is in the deep dermis. What is it doing there? It's a slowly adapting receptor. Now, if you say it is a slowly adapting receptor, fine, but what is it doing there? It is for dermal stretch. But sir, I have seen Ruffini receptor written as a thermoreceptor. Some student is telling. Yeah, okay. Maybe some author is telling it is a thermoreceptor, but I really doubt that. When somebody asks you about Ruffini receptor, the first answer should be it is a receptor for dermal stretch. Maybe if the option is not available, you can tell that Ruffini receptor is a thermoreceptor. Maybe. I don't believe it, but maybe this could be your answer. Second answer. Can you tell me the two slowly adapting receptors now? One was Merkel receptor and the other is Ruffini receptor. Can you tell me the two rapidly adapting receptor then? One was the Meissner corpuscle and the other is the Pacinian corpuscle. So now you know two rapidly adapting receptors, the meso and the Pacinian. We will discuss them in the central nervous system, neuroanatomy again. And these are the two slowly adapting receptors. What about the free nerve endings? Free nerve endings. As you are talking about the free nerve endings, you will find they are coming into the epithelium. The free nerve endings which are coming to the epithelium, they are non-myelinated axons. And if they are non-myelinated axons, that means slow conducting. Yeah, slow conducting because non-myelinated. What are they conducting the information of? So in the epithelium comes the free nerve ending, and they are non-myelinated fibers, C fibers. So C fibers, non-myelinating fibers, slowly connecting fibers. What are they conducting? Pain and temperature. Pain and temperature. So pain and temperature from the skin epithelium is carried by C fibers, that is slow connecting fiber, free nerve ending.